Manual reticulocyte count. Reticulocytes are non-nucleated red blood cells which contain ribosomes and mitochondria. A stain for either would distinguish them from mature red blood cells. Red blood cells in suspension are incubated with a supravital stain, which simultaneously denatures, aggregates, and stains ribosomes. In smears of cells stained in this manner, the reticulocytes contain multiple irregular blue strands, whereas mature red blood cells contain none. For this procedure, you will need 12 by 75 tubes, new methylene blue stain, and glass slides, disposable pipettes, a timing device, and mylar capillary tubes to facilitate making a blood smear. To stain the reticulocytes, label a 12 by 75 tube with the patient information. Mix the sample. Using a disposable pipette, add 3 drops of sample to the appropriately labeled tube. Using a different disposable pipette, add equal amounts of new methylene stain to the sample. And swirl the sample to mix. Set a timer for 10 minutes and allow the sample to incubate for that time. After the 10-minute incubation, prepare 3 slides using the wedge pull film technique. Label the slides with the appropriate patient information and allow the slides to air dry. Place the slide in the microscope using the 10x and 40x objective. Find an area where the cells are barely touching each other. Using the 100x objective, locate a single layer of cells. Begin counting red blood cells. The number of reticulated cells is determined by counting 1,000 red blood cells. Red blood cells will appear bluish-green. Count 500 cells in one slide and 500 cells on the second slide. Reticulocyte count is determined by examining consecutive fields of the smear. Reticulated cells have a dark bluish-green staining strands or granules inside of them. If you are unsure if the cells are reticulated, focus the object up and down. Artifacts will become refractive, but the reticulated inclusions will go in and out of focus. To calculate the reticulocyte percent, and if the number of red cells is 1,000, divide the number of reticulocytes by 10. Results are reported as percent reticulocytes.